Hi, everybody. This short presentation is going to give you a little background information for the protein fingerprinting lab that unfortunately we're not going to be able to do. But I just want to give you some extra information because I am going to ask you to not only listen to this presentation, but also to read about the background information in your lab book. Watch some videos that were done by the lab company that creates this lab so you could see the techniques and procedures that we would have used in order to do this lab. And even answer those discussion questions in your lab book itself so you can make sure you have a good understanding of it. And finally, you have probably seen that I've shared with you in the Google Classroom a figure, because if we would have been able to do this, we would have been able to do a figure assignment. So I have given you the figure, and I would still like you to do that figure assignment. So to give you an idea of what is going on in the lab, I have a short PowerPoint that I wanted to talk through with you. So in a minute, you'll no longer see my smiling face. Okay, now it's smiling. I think. There we go. All right, so if we would have been able to do this lab, what I would have had for you is a bunch of samples of fish that I would have picked up from the grocery store. And what we're going to try to do is to compare the different proteins that are found amongst all these different species of fish. And usually I try to pick like catfish, which would be kind of like down at the bottom, maybe salmon, that would be in freshwater, maybe some swordfish, that would be in saltwater, just so that we could see if there is a big difference in protein composition amongst different types of fish. So the study of the differences in proteins that exist amongst different cell types is called proteomics. And the interesting thing about proteomics is sometimes they have found that it could be more helpful than genomics, which is the study of DNA, because if you remember, every cell in an organism's body, except for their sex cells, every cell has the exact same DNA molecule in it. So if you were to do a genetic study on a neuron cell and a genetic study on a muscle cell, the DNA would be the same. So even though those cells look completely different from each other and do completely different things, their DNA would still be exactly the same. So sometimes it is actually more useful to look at the proteins. Because if you remember, when cells go to differentiate or to specialize, they're only using a handful of genes that are on that DNA molecule. They are only turning on or expressing the genes that they need to do their jobs. And hopefully you remember that genes are instructions for creating proteins. So if certain genes are being turned on in one cell, there's a certain types of proteins that are being made. And if other genes are being turned on in another cell, then they are creating a different set of proteins. So again, that is why it's a little bit more helpful to actually study proteomics sometimes than genomics. So I found this picture online, and it just shows a handful of different types of cells. And yet, if all these cells came from the same organism, they would all have the same DNA molecule in them. But because they are expressing different genes in that DNA molecule, they are all producing different proteins. And that's why they look differently and behave differently from each other, is because of the types of proteins they are expressing from the DNA molecule. So, in case you didn't make the connection, fish meat and fish muscle are the same thing. And different fish, obviously different species of fish, have slightly different proteins in their muscles. And that's the point of the lab, would have been to kind of compare the protein compositions. Now, one thing that you will see when you look at the gel figure is you will see that we kind of use the standard, an actin and myosin standard, last year when we did this lab. And actin and myosin are two proteins that are needed for muscle contraction. Those of you that took anatomy semester 1, we went over this right before Christmas break, how muscles contract, and hopefully you remember those actin and myosin. Hollis, you have the anatomy book, so you could go back and look at the beginning of the muscular system chapter and check out the actin and myosin. But the point is, you need actin and myosin for a muscle contraction. So therefore, all fish in their muscles, they should have those two proteins. They should have actin and myosin inside, regardless of the type of fish that they are. So that's why that is kind of a standard or kind of a control to make sure we would have actually extracted some of the proteins out of those cells. Because if they didn't come out and they weren't showing up on the gel, then we probably had an issue trying to take some of the proteins out of the cells themselves. All right, so hopefully you have a decent idea now of what that lab was supposed to entail. But I also wanted to remind you of the differences between protein and DNA electrophoresis. This is something we actually did last semester when we compared first the plasmid, and then later we actually compared the protein. I'm trying to remember what the protein was called, if that had a special name. But we did one gel with just the DNA, and then we did one gel with the actual protein in it. And so just a few quick reminders for the differences. One of them are the types of material that makes up the gel. So the protein electrophoresis uses a polyacrylamide gel. They are very thin, if you remember that from last semester, and they were pre-made. The DNA gels use agarose, and those are the ones that you've made plenty of at this point in time, and they're a lot thicker. Then, if you recall from last semester, the protein electrophoresis chamber actually runs the gel vertically. Remember, we had these very fine pipette tips to help us insert the DNA into those gels, where the DNA electrophoresis machine runs them horizontally. And yes, we had to use pipette tips, but they didn't have to be nearly as fine as they had to in the protein gel electrophoresis. And then finally, the last difference, which is pretty major, is that if you recall, DNA, because of its phosphate, has a natural negative charge. So we didn't really have to prep the DNA samples much before we ran them through the gel, besides just dyeing them. But proteins do not have a natural negative charge. In fact, all the amino acids that bond together to create them may have different charges. Some have positive charges, some have negative charges, which helps them create their weird, funky 3D shape. So what we would have had to do before we would have been able to add the proteins to the electrophoresis chamber is we would have had to treat them with a chemical called SDS. And the SDS, if you recall, number one, it gives the proteins an overall negative charge. And then now that they're kind of coated with this negative charge, the different amino acids are no longer connected to or attracted, that's the word, attracted to each other. So it actually straightens out from that 3D shape and becomes linear, so it can actually move through the gel. So hopefully that helps clear up maybe what exactly this lab was all about. Again, read the information in the lab book, answer the discussion questions in the lab book, work with the figure assignment, the gel that I gave you. Do a great figure assignment. Make sure you look at the rubric and look at the units in the lab book for the protein ladder that would be in the gel. And as always, let me know if you have any questions. Hope this makes sense. See you guys later. Bye.